All right, new chapter, chapter 14. So, almost for sure you've heard about this type of biomolecule, um, probably with no exceptions. And whether it be in a biology class or whether we're talking about consuming protein, excuse me, or nutrition, things like that. You've almost certainly heard of proteins. So the idea is that these are also biomolecules. Many of them are polymeric, I guess is what I was going to say. And their monomer unit, the fundamental unit that makes them up, is called the amino acid, and that's kind of where we start. We just kind of like with carbohydrates, we started with monosaccharides, excuse me, and worked up to disaccharides and oligosaccharides, and then we talked about polysaccharides. In the same way, we'll start out with the monomer unit, excuse me, which is the amino acid. So we'll get to that here in a second. But let's look at it. There's just some factoids, some interesting information, and I do want you to kind of generally know a lot of this stuff that we'll go through. Because it is important. So the idea here with this slide is that proteins make up a large cross section, a large number of the biomolecules that exist in living organisms are in this category, exist in this category. And part of the reason for that too is we'll talk about enzymes, excuse me, which are proteins also. They're just catalytic proteins, proteins with a function. So they kind of fit into this category as well. And there's tons of different kinds of enzymes as we'll see. So anyway, now there's there's tons of different ways that you can categorize proteins, but you see one of these ways, I should say, is based on their function. So let's look at an example of each of these different types of proteins, or these different classes. So structural proteins. So for example, the one I always think about is keratin, because it's in hair and nails. I almost think we've talked about that before. We talked about nail hardeners, I think, yeah, in our when we talked about formaldehyde and how it's in some of these nail hardeners. But keratin, so I'll just put in, hair and fingernails. Okay. Catalyst. Okay, so catalyst, again, we won't belabor this one. We'll talk all about all sorts of specific enzymes later on. But let's just put that. Let's just put enzymes. Enzymes are are proteins. Okay, and they're catalysts, or biological. Biological catalysts. Okay. Movement. Okay, so the one I'm thinking about here, or the couple I'm thinking of here, are already on the slide here. So actin and myosin. These are, again, we're just giving examples. Yeah, so actin and myosin. Okay, these function in movement, muscle contraction, and if you haven't studied that in a biology class before, almost certainly you will at some point along the way. Again, you don't need to know right now the specifics of related to how muscles contract and how actin and myosin function in that process. We're just giving examples of each. Okay, so transport. Lots of possibilities. I'll just put hemoglobin functions in O2 transport. Okay, and we've also transport. I guess that's okay. And we've also talked about transmembrane proteins. So transmembrane proteins, right? In fact, we just saw some of those in those lipoprotein particles. Right, and so that I think it was that complex lipid monolayer had in it transmembrane proteins. Things can diffuse through those. So again, they can cross cell membranes. So transport different signaling molecules, and a number of possible things be transported across membranes that way. Okay, hormones is the next one. We talked about hormones a lot too in our lipids chapter, but some examples I gave you, I'm trying to think like back to that negative feedback pathway. Whoops. So GnRH, gonadotropin releasing hormone, luteinizing hormone, follicle stimulating hormone. These were in that negative feedback pathway, if I remember right. And these are hormones, and more specifically, they are what we call peptide hormones. Okay, so let me just write that, peptide hormones. Okay, and the significance of that is, remember, we talked about steroid hormones also, but steroid hormones those are the ones that were synthesized from cholesterol. They have that tetracyclic ring system. These are not, these are protein hormones, or what we'll see is peptides are small proteins. So anyway, okay. So protection. So let's think about an example I often give is these two. So fibrin, fibrinogen are proteins that function in clotting, so blood clotting, and that certainly is a sort of a protective mechanism for us. So there's some examples there. Storage. I think we have one of these upcoming slides. Ferritin is an example of a storage protein. It stores iron. Okay, so we'll see that that's a sort of an important storage protein. Gosh, regulation, that's a broad category. But if we think about polymerases, so DNA polymerase would be a great example. This is actually an enzyme, but again, all enzymes are proteins. So DNA polymerase functions in DNA synthesis and things like that. So, and of course, DNA synthesis DNA regulates production of RNA and proteins and all those sorts of things. So definitely a regulatory process. Now, if we're thinking about just what's left on that slide, it's, again, there's so many ways of classifying or categorizing these things, but generally speaking, excuse me, we do have proteins that kind of fold up and are more globular in nature, and some that are more linear or fibrous. So it's kind of like back to our carbohydrates chapter, and we're thinking about the like starch, the different forms of starch. So amylose and amylopectin. And if you'll remember, amylose was more linear because it didn't have those branching points, and amylopectin was more branched. And of course, if you looked at it under a scanning electron microscope, it would seem more spherical or circular or globular, whereas the amylose sort of aggregated into these more linear constructs. And it's not exactly like that here, meaning we don't have, like if we think about the polymeric form of a protein, it's not a branched form, but again, it's how these things aggregate or how they fold up, which dictates kind of what category they belong in, whether fibrous or globular. So, okay, here's another. Again, we throughout this chapter we look at different examples, and of course we couldn't look at. We'd spend an hour looking at all those proteins we just mentioned, right? But this one, I suppose, right? Doesn't hurt to just highlight one of them. So ferritin, and I think we come back to this later, like in our nutrition chapter when we talk about the importance of iron in our diets. But the idea is here, right? In fact, you see this here. You have this protein is it's an interesting protein. We can use it as an example later too, because it has what we call subunits. So there's different levels of protein structure, but basically there are 24 separate proteins that come together and aggregate together in what we call a quaternary level of protein structure, and they create this big spherical large protein essentially. But inside the protein there are all these iron ions, and it's one of the ways we store iron. In fact, we can't. The only way we get iron is from our diet. In fact, and you see up here on the sort of the top right here, we absorb a certain amount, couple of milligrams a day, but we lose that same amount, and so we do need it in our diets. Most of it goes to making essentially heme or hemoglobin, again, which is important for oxygen transport, and that happens in our bones. But you see that this 10 to 20% of it goes towards storage. We store it for a rainy day, right? So anyway, so now, okay, so this slide is meant to show us, okay, we have these individual, we've said this, but we haven't encountered it yet. We have these individual monomeric units called amino acids. There are 20 naturally occurring ones, and we'll look at those. But those are the ones that come together to form a polypeptide chain, or what we will refer to as a protein. So we'll start out with the amino acids, and then we'll build on this later, and we'll start to write out peptides and proteins, draw them out, form them, and so on. So, okay, so first things first, we often call amino acids alpha amino acids. There are other types, but the ones we're referring to as forming these proteins, these naturally occurring proteins that we're talking about, are the alpha amino acids. Okay, so some of this is on the slide, but let's do this kind of like we did before. Anatomy of this, anatomy of that. Let's do. Whoops, that's not what I wanted. Right here. So anatomy of an alpha amino acid. Anatomy of an alpha amino acid. So. Okay, so I'm just going to write this, and there's a lot of this needs explaining, but these have what we call an amino end and a carboxyl end, or an amino end and a carboxylic acid end. Thankfully, we know exactly what those groups are, right? So here's the amino or the amino end, and there's the carboxylic acid, and then there's an R, what we call an R group. Okay, so let's do this. And first, okay. So, oh man, what happened there? There we go. Okay, so this, by the way, is the alpha carbon. Now, if you'll remember, right, this is like an aside. Maybe that's not large enough. Like this is like an aside. Remember when we talked about keto-enol tautomerism, right, in a previous chapter, and we said that anytime you have a carbonyl, right, like let's say, well. Okay, let's do this. Anytime you have a carbonyl, the carbon right next, so the carbon or carbons, there's two of them here, right next to the carbonyl are referred to as alpha carbons. So take a look, right? We have our carboxylic acid, but more specifically, there's the carbonyl, and the carbon right next door is the alpha carbon. So that's where the alpha in alpha amino acid comes from, because check it out, this is our amino group, right? Amino, I'll just put FG for functional group, right? We've seen that enough times. Functional group, okay? And so it's an alpha amino acid, meaning, okay, the amino group is attached to the alpha carbon, and so is the acid. So there's the acid, right? There's the carboxylic acid. So both things, right, the amino group and the acid group are attached to the alpha carbon of the amino acid. Now, the R group here is I'll just put the basis for classification of the 20 different naturally occurring amino acids. Okay, so what I mean by that is they're different, right? So each amino acid, I'll just put AA if you don't mind for amino acids from now on, has a different R group. So this is a good chance for us to, whoops, let me get rid of that. This is a good chance for us to explore those a little bit. So I think I have these, yeah, I pulled these up. No, this is what I wanted. Yeah. Now I have a couple of different, I can post these as well. And we're going to talk about different forms of an amino acid, whether charged or uncharged at different pHs. But one of the things you see is, right, a certain part of these, right, is exactly the same for every one, meaning there's the amino group. Notice the nitrogen has an extra hydrogen. We'll talk about that. There's the carboxyl group. There's the alpha carbon. There's the hydrogen, right, attached to the carbon, which we know is there, right? It's implied in the structure we drew. But notice in this box, this is the functional group, the R group that makes leucine. Notice it's obviously different for alanine. It's just a methyl group for alanine. For valine, it's an isopropyl group, right? And so there are 20 naturally occurring amino acids, and it's also the basis for, even within the amino acids, they're often classified as, and you see it in this sheet, as non-polar hydrophobic. You've got some polar R groups. You've got some acidic R groups. You've got basic R groups, and you can you've got aromatic ones. So anyway, that's what I'm getting at here, is that the R group is different for all of these amino acids. Notice here's another here's just another sheet that gives us the amino acids. It just looks a little different than the first. And again, I want to go back because I'm saying a few of these things, but I want to actually highlight something. Okay, so oftentimes amino acids are written different ways. So this is what we call the uncharged or neutral. Let me go back to here for a second. Back here. So this is uncharged or neutral form of an amino acid. Okay, but here's another possibility. We know that amino groups, right, are basic, right? In fact, they're the quintessential base. Amines are. So nitrogen can, if there's hydrogens around, gain an extra hydrogen. In which case, I'm going to redraw this. Just keeps bouncing back and forth. I'm going to redraw this now as NH3, and then we'll put that same R group there. And then I'm going to draw the carboxyl group as lacking a hydrogen, because same idea, carboxylic acids, right? They're the quintessential organic acid. And we know that acids can do what? Give up H, right? Donate H. So this, and I should put a positive charge there, this is what we refer to as, I was trying to separate these. I'll just put versus, right? VS. So this is what we refer to as the zwitterionic form. Now, let's talk about, I think I have it on that last slide, but let's just talk really quickly about what a zwitterion is. And it's like a German word. Okay. So zwitterion means, sometimes, I think this is probably there too, sometimes we call it an internal salt. And if you remember, a salt is an ionic compound, right? So the idea here is a zwitterion possesses both. This is driving me nuts. I'm sorry, give me 1 second here. I've got the table I'm writing on is just like, it's got a wobbly leg. Give me a sec. Worse. Sorry, give me a sec. Okay, much better. Okay, so zwitterion possesses both positive and negative charges within the same molecule. I'll just put within the same compound. Okay, I'll just put overall a zwitterion is neutral. So notice we've got the positive charge and the negative charge within the same molecule. So overall, it's neutral. Okay. Now it is also possible. So I'll just put this down here. Also possible to have these forms. And we'll go into this later. It's a pH dependent thing. I'll just put depending upon pH. So we'll go into this later. But you can think about a situation where you have the nitrogen protonated and the carboxyl group protonated, or you have the nitrogen deprotonated, just the regular amine, and you have the carboxyl group deprotonated. So those are both possibilities as well. So don't be surprised if you encounter any of these. And this is one of the things that I was getting at with those amino acid handouts. By the way, so I don't forget to mention this, you do not have to memorize the amino acids. They will be given to you either in a particular problem if you need them, or like on an exam you would have the sheet of amino acids excuse me, attached to your exam. So, but I guess what I mean to say is don't be surprised to see any of these different forms of a particular amino acid written out. Okay, sometimes there's a preference for one or the other. Sometimes there's a reason we draw one or the other. So anyway, just don't be surprised to see them. Okay. Now next thing, and you're probably, you look at the top of the slide, right? You look at the title of the slide, and you're just like, oh my goodness, right? I thought I had for once and for all left this behind, but it turns out amino acids and of course then proteins that they're made from are chiral. In fact, all of the naturally occurring amino acids, except for one, glycine, they're all chiral. A couple of them even have a couple of chiral centers. So, and you can probably go back to this, and if you think about this for a second, right? Let's just think about this. These are generic, right? We haven't talked about it. Well, we've seen some, but this is generic, right? We haven't named the R group. But if you look at this alpha carbon right here, notice 1, 2, 3, and then of course we know there's a hydrogen there as well. 4 different things. So no surprise, these have chirality associated with them. They are chiral compounds. Okay, back to this. Okay, so now, well, one of the things I was just gonna get to though is we've used RNS, right, as absolute configuration of a chiral center. We know how to do that Kahn-Ingold-Prelog system, but again, you're gonna roll your eyes when I tell you this. We use a different convention really for determining stereochemistry of an amino acid rather than RNS. Kind of like with monosaccharides, we use D and L rather. And again, it's really annoying. I wish we just use RRS, but it's been done this way forever. So you see a picture there of L-alanine. So we're going to do this together. And I think I even have a couple of problems. I think I had that worksheet. Hold on. I think I pulled up a worksheet here. Yeah. Okay. So you see well, I should probably draw this in the slide so that we're doing it there too. But a couple things there for part A. Draw the zwitterionic form of alanine as written, and then determine if this version of alanine is an L or a D amino acid. So let me actually reproduce that on the slide if I can remember it. So it's this. Whoops. Okay, I think it was this. I'm hoping I drew that right. And I'm just drawing the neutral form. Again, doesn't matter. We could draw the zwitterionic form, wouldn't matter. And let me just say this too, right? This is what we drew, right? This would be alanine, CH3. Sorry, it's so far up there. But the way we just drew this, right, it's a two-dimensional rendering, right? But we know that these molecules don't exist in 2 dimensions. So, this is a 3-dimensional version of alanine. Notice the groups are all the same. We've just written it a little differently so that, again, with a dash and a wedge, like we're used to when we draw 3-dimensional compounds. Now, if we want to determine D or L, now we could easily determine RRS, but again, that's not generally speaking what we do. I suppose we could just make it, after we're all done, if we remember, we could say, oh yeah, it's R, or oh yeah, it's S. But okay, let's do this. Here, I'm gonna write the method. So let's put method for determining D, or whether an amino acid. Let's say that. Sorry, hold on. I'm gonna grab a drink real quick. Whether amino acid is D or L. So let's do this first, okay? And a lot of this will look familiar to you. So convert 3D molecular representation to Fisher projection. Okay, we've done that quite a bit by now, so that's good news. Okay, we can do that, I think. So number 2, using the rules we know. So let's just put using. This is so sensitive. The rules for let's say rotating or manipulating. Let's say that manipulating Fisher projections. Okay, so let's put place carboxyl group COOH, or depending, right? COO minus, I suppose, like we just talked about. COO minus up, okay, and the R group down. Okay. Now, if we do that, if and when we do that, let's just say if the amino group, and again that's the NH2, or depending upon the NH3 plus group, amino group is pointed to the right, the amino acid is a D. If it's pointed to the left, the amino acid is L. Okay, so D or L. Now I want to remind you of when we did this exact thing with the monosaccharides. Remember, we were looking at the position of the OH in a Fisher projection, the position of the OH group, whether it was pointed to the right or to the left, right? And it was the OH group specifically on the penultimate carbon. Here it's the amino group on the alpha carbon. Okay, so just so we're clear. All right, so let's actually do this. Excuse me. With the example we're given. And I guess if I remember back to that little worksheet, we also wanted to draw the zwitterionic form. Maybe we should just, well, yeah, I guess we could do that at the end. Okay, so remember, there's a lot of ways we can do this, but what we're trying to do, remember, so we're just going to put, I'll just put rotate, because that's really what we're doing here. Rotate this such that, remember, it's the bow tie, right? It's the 2 wedges and then the 2 dashes. Sorry, let me make this bigger and a little nicer. I don't know if that's any better. Okay, so there we go. We got the bow tie going. Okay, and I always think the easiest way of doing it, like when it's written the way it's written, is to just kind of open it up and face it towards you. So if I do that, I wish we were together right now, I would bring out a model, right? And you can always do that for yourself if you'd like. But then if we do that, if we kind of just open it up, the CH3 is actually still pointed up and the H then is pointed down. Now remember, what we do next is smash this, right? Just like push it into 2 dimensions, and what we're left with then is our Fisher projection. Everything is in exactly the same place that we have it now. So there's our COO. Okay, now, what we have right now is well, the amino group is pointed to the right, and so we're like looking at that and we're like, okay, can we just jump to rule 3 and are we already ready to label it as DRL? But remember, this right here is the R group, and this right here is the carboxylic acid or carboxyl group. So the carboxyl group needs to actually be up, and the R group needs to be down. Now remember, we've got a lot of ways we could, first of all, have even made this Fisher projection, but then we have a lot of freedom. But I'm gonna grab green here. We're gonna hold one group and rotate the others clockwise or counterclockwise. Now you could do multiple rotations. I think we could even get by with just one rotation here. You may be thinking the same thing I am. I'm thinking, why don't I just hold the amino group, and I'm gonna then rotate. Then I think I can do this all at once, right? Because I want the carboxyl group up and the R group down. So if I just hold the amino group, gosh, then in that case, and we'll do another practice problem here as well. Right? This one's kind of anticlimactic. It wasn't too bad. So in that case, right, what I end up with is a Fisher projection where CH3 is here. I'm running out of room here. H is here. COOH is here, and the NH2 is there, right? And so obviously, right, we've got our amino group pointed to the right, with the other things being true, right? The other stipulations have been accounted for or been, we've checked those boxes, and so we're going to call this a D amino acid. So, and we actually call it D-alanine in this case. Now, if we wanted to draw the zwitterionic form of alanine, I'm just going to go back to I guess we could do it in the I don't have a lot of extra room here, but let me just write zwitter. Whoops. The zwitterion. I'm just going to go back to the three-dimensional form. Remember, it's the two ionizable sites are the amino group and the carboxyl group. And the only way you can ionize an amino group, right, is to add, right, it's a base, you add an H to it. And then the way that you would do that to a carboxyl group is for it to lose an H. So the zwitterionic form would look like this. And I guess we hadn't stipulated that it had to be, gosh, I guess we could have done 3 possibilities, right? We could have done this one as well. And it kind of just depends on what you're asked to do. We could have done this, right? This is that 2-dimensional form, or we could have even, I suppose, done it in kind of the way we drew it in a Fisher projection form. All of these, right, are these internal salt forms, or the where we have a plus and a minus. I should make sure we put the plus there. Oh shoot, you're looking at me and I'm just like, what are you talking about? You didn't put the positive, right? So I gotta make sure we got that down for all of them. Positive and a negative. Okay, so these are zwitterions, zwitterionic forms. Okay, of alanine, just drawn different ways. Okay. Now let me go back and look at, just because I imagine you maybe like wouldn't hurt having an extra problem to do. And if we do, let me actually get another slide there, a blank one. There. Okay, where was I? Just lost track of. Yeah. Okay. So there was another one there, and zwitterionic form of valine, and then determine if, as written, it's DRL. So let's go back. I think I can remember this one. Okay. So let's. think it was the neutral form that was written. Anyway, this is valine. Okay, now, zwitterionic form, I'll just draw the three-dimensional one, just as written. Again, the whole idea here is we put an extra H on nitrogen. All right, we've done this a number of times now, but I keep not doing it. So H3N, okay, O minus, and then, okay, and then the R group, which is that. Okay, now let's figure out if this is D or L. Now I'm gonna, again, we want to do this, so I'm just gonna even draw it out before we even look at it carefully, right? Because this is what I want. I want to be able to put the molecule into this form. Now, it's written just a little differently than the other one. I'm just going to put our H here just to remind us that there is an H there. You know, it doesn't, I mean, you wouldn't have to do that if you're able to just kind of rapid fire draw it out. But it's just a helpful reminder. And I'm going to actually think about, just kind of like, gosh, again, this is where like me having a model and us being together would be helpful. And there's so many possibilities here. Actually, let me just tweak it one way. So if I were to put this to the left and hydrogen to the right, I'll bet you could see that I had kind of twisted it down and to the right and then made it face me a little bit. Such that I would have an NH2 pointing up and my carboxyl group pointed down. I'm just going to write my carboxyl group, excuse me, COOH, just to make it easier on myself. Okay, now I take that, right, and remember I smash it, and so I end up with my Fisher projection, everything where it is. Remember, it's 2 dimensions now, so I don't need any dashes or wedges, but I will, right? There's my V, or my valine. That's how I think about valine. The isopropyl group looks like a V, but that's how I remember valine. Excuse me, I'll grab a drink here. Okay, so next thing, remember, we wrote this down on the last slide. I want my carboxyl group faced up, and I want my R group face down. Okay, and then wherever the amino group ends up, it ends up. But okay, so I might have to do a couple of manipulations here. I'm trying to think. Could I get by with? I think no, because carboxyl group has to go up. So I'm gonna have to do a couple of manipulations. So let's do. Doesn't really matter. Let's hold. I'll get this green one here. So we want to put this up. Let's just hold valine where where it is, and let's rotate right clockwise. Could go either way, but clockwise in this case is what we want because we want carboxyl group up. So let's redraw this. COOH, and then NH2 here, and then H there. Right, and then my valine stays the same. Did I do that right? Yeah, it looks good. Okay, now I've got to do one more. And so in this case, right, I want my R group down. So I'm just going to keep, because remember I got to rotate 3. I'm definitely going to keep my carboxyl group where it is, right? Because that I need to have up. So let's leave that one there. And then let's rotate the other ones. And I guess I got to rotate them this way, right? Because I've got to have my R group down. So I'm only left really with one choice here. So if I redraw this, I have hydrogen and my COOH is up. My valine, or my R group, my isopropyl group is down. And did I get that right? Yeah, NH2 is here. Across, down, up. Yeah, okay. So the idea here is that now I have made, and I'm hoping you'll agree with me, right? My carboxyl group's up, my R group's down, but now NH2 is pointed to the left, right? So pointed to the left. So this is not D anymore, it's L valine. And let's just be clear, this is also L-valine, right? It was originally valine, but we didn't know if it was D or L. And I should say it's really the three-dimensional one that's the L one, right? Because that's the one that reflects stereochemistry. But I wrote it down here as well as L-valine. Okay. Okay. So let's see what's next. All right. So I think these are just probably from our slides. It's basically just what I have on those handouts. It's all of the different amino acids put into different categories, whether they be polar or non-polar. Acidic or basic. This is kind of cool. This shows you those two exceptions. That should actually be an alpha there. Okay, I should say alpha. But it shows you that these two exceptions, because they have actually a pair of, if you think about it, stereocenters, two of them. All the other ones have one, and then glycine has none. Glycine, by the way, I have mentioned it a couple times. Let me just show it to you. Glycine, its R group is H, so there are two H's attached. There it is, right there. There are two H's attached to the alpha carbon, so it's not chiral. Okay, now we've been talking about zwitterions, right? And I want to go back. I just want to go back. For some reason, I was thinking maybe I missed something. No, okay, I think we got it all. Okay, so we've talked about zwitterionic forms of. Gosh, did we write the zwitterion? No, I'm just. Yeah, we did. Okay, I just couldn't remember if we'd written the zwitterionic form of valine, but we did. Okay, so we've talked about zwitterionic forms of amino acids. We'll continue to talk about those. By the way, peptides and proteins can also have zwitterionic forms, something that we'll get to here in just a little bit. But there's something called an isoelectric point for every single amino acid, and it's different if you look at this table. It's true also of proteins. They have different isoelectric points. And by the way, the isoelectric point is the pH at which, and it was in the last slide there if you want to just refer back to it. I guess I can write it here. PI, or isoelectric point. Just so we can refer to it while we're talking about this on this slide. Isoelectric point is the pH at which the amino acid, I'll just put or peptide or protein because it can be applied to that as well. Apply to those as well, or peptide or protein. Primarily, i.e. most of it, exists in, its zwitterionic form. So most of it exists in its zwitterionic form. So now, I was alluding to this earlier. I said this earlier. Whether an amino acid, and it's true of proteins and peptides too, but whether one is charged or uncharged or in its zwitterionic form or in its neutral form is really a pH dependent thing. And so let's elaborate a little bit on that. I think we have the tools to kind of understand this. But let's say we start out with, and let's just do this generically. So we don't care what amino acid this is. So let's just say it's some, drop my pen. Some, excuse me, no, OH. Okay, we'll just call it the uncharged form of a random, or not random, generic amino acid. That's why I put R there, okay? Okay, so let's say we lower the pH, or we could just put at low pH, really low pH, let's say very low pH. Right, what's true about a low pH environment? And all of us, right, we've been through our acids and bases chapter, certainly in this class and also in Chem 3A, we know that at low pH, right, below 7, there's an abundance or an excess of H+, of hydrogen ion. So we have what we could say here is just to remind ourselves of that. We're talking about acidic environment. So an acidic environment is characterized by lots of H+. Okay, acidic environment, lots of H+. So, excuse me. well, I would ask you, okay, what are the implications of that? Remember, these amino acids have an amine group, and it's the amine group that can act as a base and latch on to an H, right? And so the idea here is that we would expect then at low pH, right? And we could write that, if well, I think we've been through this, and I don't think we necessarily need to. Nitrogen, right? If you want to write this as an aside, right, you could write that lone pair of electrons on nitrogen latching on to, an H plus or whatever, just to remind yourselves that that's what amines do. But what we would end up with is the positively charged form. Now, nothing happened to the carboxyl group. You can't doubly protonate a carboxyl group, right? So anyway, you would have then the positively charged form, okay? Now, if we take that same amino acid, that generic one, and we say, okay, let's, instead of adding a bunch of acid, let's add a bunch of base. So at very high pH, right, and of course, basic environment, excuse me. So basic environment is characterized, by lots of OH minus. So OH minus, right, is our quintessential base. It grabs H pluses, right? Now, it can't really grab one from the amine. I suppose it could. It doesn't in this case. Amines don't easily lose, I should say, neutral amino groups don't easily lose their H's. It's not that they can't, but it's rare, especially in situations that we've discussed. So really the only H plus that's likely to come off is from something that's relatively acidic, like a carboxyl group, right? And so you see where this is going, I think. R group there, and now I have my carboxyl group that has become, again, something we've discussed, a carboxylate group, right? And I suppose we could write plus water, but that's beside the point, right? So this is the negatively charged form. So we have a positively charged form of an amino acid at really low pH, a negatively charged form at really high pH, but then there's somewhere in between. Now, it's not always at pH 7. A lot of times we would guess that it's at pH 7. It's not always true for reasons we won't get into, but there is a pH at which, so we'll just put at its isoelectric point, okay, most or all, I'll just put most of, it's never all, but so most of this amino acid exists, right, we've said this several times, but okay, so I'll just say as this compound, right? Plus, don't forget the plus. Okay, of course this is the zwitterionic form, okay, of this generic amino acid. Okay, so, and we're going to apply this same methodology to proteins, and this has implications with respect to physical properties, things like solubility and stuff like that. So we'll get to that here in just a little bit as well. Okay, now, some more, I guess, well, and this stuff's really, it's not only interesting, from a just because there's some interesting applications that I really do want you to kind of know and latch on to, but we'll talk more about disulfides later on as a stabilizing influence when we're talking about how proteins are all folded up and stuff like that. But it's what's interesting about cysteine, and again, we're going to highlight a few separate amino acids and just talk about some things that they do and that they're involved in. But cysteine is one of those that's important because it forms what we call disulfide bridges. Now, if we get our laser pointer here, okay, so what we're seeing is two separate cysteines. And by the way, they're shown as just individual amino acids, but it is true that these cysteines could be part of proteins, and this same thing would happen. Again, more to come on that later. But if you're thinking about this reaction, and it is a reaction actually, right? Two of these coming together, and forming this disulfide bridge, and there's a loss of hydrogen here, and that isn't really shown as a byproduct or anything, but what you see is if you're going in this direction, we have an oxidation of cysteine, right? And we form, it's called cysteine, but this disulfide, what we call a disulfide bridge. Now, just a reminder, why is this an oxidation going in from top to bottom? Remember, oxidation refers to either or, right? So either the increase in oxygen of something, right? Something increases in the amount of oxygen it possesses, or decrease in hydrogen, right? And the opposite reduction is an increase in hydrogen and or a decrease in oxygen. And really, it's not oxygen in this case that's being gained or lost, it's hydrogen, right? So in going from top to bottom, you have a loss of hydrogen, so an oxidation, in going from bottom to top. So that that disulfide bond were to be what we call reduced, it would then, hydrogen would be added to it, and you would reform the individual cysteine thiol functional groups. SH groups are thiols, and I would say remember, but we skipped thiols. Remember, so it is something we should know is what a thiol group is. So a thiol group just generally is, and this is good. This is good for us to do here. Just, I think I probably at the very beginning of our organic chemistry chapter, I think I put this in like when we were like when we first started talking about OChem. Like I think when we wrote out all those functional groups, I think I wrote these, but it's been a long time, right? So this is a thiol, and then this is a disulfide. Okay, and that's what you see here. Okay. Again, more to come on these as we talk about proteins. Now, some kind of interesting application stuff here. It turns out that one of the things, there's a couple, but one of the things that makes hair more or less curly is how many disulfide bridges it has in it. So, you know, if a person has a lot of cysteine in their hair and those cysteines come together and are oxidized and they form disulfide bridges, what can happen is that like contorts these what we call alpha helices in the keratin in your hair. That's the protein in your hair, and you end up with more curls or more waves. And so, but that can be artificially, I guess, altered. You know, if you go to the, I never know like what to. So beautician, or if you go to the hairdresser or what. I'm not sure what the word is, but anyway, that if you go to a person and you want like, and this is the example here is a perm. I don't know if anyone's ever had a perm. I have not, but if you have, what the idea is, you break those. So if you go back to the disulfides, you actually break them. Okay, let's think about this for a second. I think I have a slide here. So if you think about a hair strand, right? Let's just think about and we're going to talk more about this. This is going to be kind of really simplified, but if you have, again, what we're talking about is these disulfide bridges hold these strands of protein, these keratin strands together. Okay, now what you can do is you can actually there's reducing agents and oxidizing agents. I think the reducing agent they use in some of these processes is called like ammonium thioglycolate, I think is what it's called. But if you do this reduction, right, we were just showing this, right now what you have is the hair, or just, oh sorry, these are just bad alpha helices. But the idea is now you have all these thiols that are just sticking out because, again, the disulfide bonds have broken, and you have SH groups now sticking out. Okay, but then, so here's what happens next is you put, and again, this is like what happens when you, not by experience, I've just heard, right? If you get a perm, right, you put your hair in rollers. So what happens is and I'm not a great artist here, but you'll hopefully get the idea. Yeah. So if you put the hair strand like you know, on a roller here, so again you still have these, but now you've made them, you've kind of artificially made them curl around, and you're going to reform. So I should put oxidation, so rollers plus oxidizing agent, or I'll just put oxidation. This is usually peroxide, like a dilute solution of peroxide. So disulfide bonds are reformed. Okay, and again, I wish I had just, my drawing is just so terrible, but now we have these SS bonds again that kind of hold these strands together. And then you get the idea, I hope. So anyway, kind of interesting, just application, right? And so, and the idea is those disulfide bonds are covalent bonds, and they don't easily break. And in fact, a perm is meant to last for a while. In fact, I think the word, right, is permanent. So it's meant to be permanent, and of course it's not totally permanent. It's permanent until your hair grows out, and I think it does probably over time get less curly and less wavy. Again, I haven't had one, but I think that's probably true. Some of you maybe could enlighten me about this stuff, but anyway. So, and again, more on disulfide bridges when we talk about proteins, how they are folded and that kind of thing. Okay. Excuse me, I'm gonna grab another drink. Okay, so a couple of other amino acids, and these are cool too because. So tyrosine and phenylalanine, something we haven't gotten into. In fact, since I'm thinking about it, now's a good time. I just am reminded of it, so I just want to say it before I forget. If we come back to one of these handouts that has all 20 naturally occurring amino acids on it, I want you to notice something. Let's just look at the top at the top amino acid. It's called leucine, okay? But it also has what we call a 3-letter abbreviation and a 1-letter abbreviation. Now, that's just a convenience thing. Later on, when we draw these big long chains of amino acids, it's just convenient to write the 3 or the 1-letter abbreviations, just so we don't have to write the name out for all of these when we're talking about a sequence. So it's just something I want you to know, that there is that, and don't be surprised. And again, if you need those, I'll give them to you. In fact, likely I will give you that handout that we were just on, and which has all 3 of them on it. So you see here, it's TYR and PHE. That's what, but I said tyrosine and phenylalanine, right? That's where those come from. Okay. Now one of the cool things, and I think I have this. So I'm gonna spoil it for you right away. But if you remember, let me get the laser pointer. Do you remember this collection of atoms arranged? So it was, I think, in our amines chapter, we talked about phenylethylamines or phenethylamines, and we also talked about tryptamines and other, I guess, biologically active amines. What's really a really interesting connection, and it's one of those things that where we often look to natural sources for inspiration or for information. So we could predict, right? So methamphetamine, right? And it turns out that these neurotransmitters that we're going to talk about, and these amino acids have been biologically active long before methamphetamine was ever dreamed up or synthesized. And so, but it is interesting to know, right? These are biologically, phenylalanine, dopamine, and L-dopa epinephrine. These are all biologically active molecules, and I should say, especially biologically active are the dopamine and the epinephrine, and they are derived from phenylalanine. And these are all in that same class of molecules as methamphetamine. And you remember MDMA, ecstasy, right? Some of these other compounds that were very similar and had that phenethylamine group of atoms incorporated into them. Just kind of interesting. These and I think I have actually. Well, yeah. Okay. So one other thing I sometimes will mention and wanted to mention was L-DOPA is also a precursor for dopamine. If you see, all that happens is there's something called decarboxylation. You lose a CO2. And there's different ways this can happen, and you end up with dopamine. But what's interesting here is that, well, you may know this from a biology class. Dopamine is one of these neurotransmitters that helps with things like muscle function, and I think they say like behavior that's reward motivated. Dopamine is functions in those neural and biological processes in the brain. But one of the things that's sad, interesting to note, also a sad thing, is that if you've ever heard of the disease, Parkinson's disease, it turns out that people that have this disease, their neurons, the ones in their brain that are releasing the dopamine, are basically degenerate over time. They are slowly destroyed. But one of the neat things, neat discoveries in science, and I think there was like a Robin Williams movie, I can't remember the name of it, that was made about this, but L-DOPA can actually be synthesized in the lab, so can dopamine. But the interesting thing here is that you'd think, oh man, we can just give a patient dopamine, right? Because they're a Parkinson's disease patient, because those neurons that make dopamine are slowly being destroyed. So let's just give them some dopamine because we can make it, right? It's not that hard to make. Well, the problem with that is dopamine itself doesn't cross into the brain. There's this thing called the blood-brain barrier. It doesn't cross into that easily, or over that into the brain easily, but a precursor, L-DOPA, does. It's more polar, it's got that carboxyl group, and there are reasons for that we won't go into, but that was a really important discovery. Now, it's not that it totally cures Parkinson's, it doesn't, but it certainly can provide an additional quality of life and some of these types of things. So just a really kind of, we won't look at that in any more detail, but it's just an interesting aside, because what's cool is you do see like the synthetic pathway all the way from phenylalanine to dopamine, and then even to epinephrine, which is another one of these neurotransmitters that a good chance you've heard of it. So, okay, what's next? I think I have another one of these. Oh, I gotta go back to the pen. Oh yeah, okay. So tryptophan is another of these naturally occurring amino acids, and I think there's this. I won't call it a misnomer because there's some truth to it, but you may have heard of this. Okay, so if we're talking about Thanksgiving here, right? And it's just like, okay, turkey has tryptophan. So you may have heard turkey makes you sleepy, or you may have also heard, well, it's the tryptophan that makes you sleepy because turkey has that in it. And one of the interesting things, and before we continue to follow that rabbit hole, let me just show you. I don't know if I have the same type of thing here on this slide where I show a tryptamine. I don't think I do. But one of the things you'll notice is, or do I? Nope, I don't. But you'll notice that same collection of atoms that's in some of those tryptamines that we studied in our amines chapter is here in tryptophan. So again, you see these, you look at them, you're like, oh, okay, that makes sense why these are biologically active. And you look down, you start with tryptophan, but same thing, tryptophan is a precursor for the biosynthesis of serotonin, which is another one of these neurotransmitters that is released into your brain by neurons in your brain. And I think this is one of those ones that's like, I always think relaxation or feelings of like well-being come from serotonin when it gets released. So serotonin, you're like, okay, well that makes sense. The tryptophan then gets converted to that, and that kind of makes us feel like we're good. We're sort of fat and sassy. We're feeling good after this huge meal at Thanksgiving. But back to the question, is it really the turkey or is it really the tryptophan that ends up making us sleepy? And the answer is, well, kind of. It turns out that there's tryptophan in lots of different things. There's tryptophan in, well, certainly turkey, but also like in chicken, in cheese, in, I can't remember all of the things. There's a number of other things. Yogurt, I think. So some of these dairy products have tryptophan in them as well. And so you're like, well, why don't all those things make me sleepy in the same way? And there's this interesting thing that happens. Tryptophan, if you look at it and compare it to some of the other amino acids, it's kind of big and bulky. And so what happens is, in particular, like at Thanksgiving meals, right, we often also eat, in addition to this turkey, right, with the tryptophan in it, we also eat often sweet things. And so that induces, well, the release of insulin, right? So insulin helps sugars go into our cells, but there's a whole bunch of other amino acids and biomolecules that are involved in that process. So what originally happens is, well, tryptophan's kind of having a hard time getting across the blood-brain barrier. It can, but there are other things that go across quicker, other amino acids that are smaller and more easily able to cross the blood-brain barrier. So it's kind of like, if we're thinking about a line, it's kind of like last in line, or one of the last amino acids that's able to get across. It's because it's got competition. But when this insulin gets secreted, there's this recruitment of a lot of these other amino acids to the cells and to these other processes that are involved in the metabolism of these sugars. And so tryptophan isn't one of those. It's just kind of left on its own. It's like, okay, now I can finally get across. It's less crowded. I can get across the blood-brain barrier. Then it does, and then it gets converted to serotonin. So the reason I kind of put the conclusion here is that it's not, if I get my pointer, it's not necessarily just the tryptophan, but if you add in, I meant to get a pointer here, but if you add in, right, some sugar, or I suppose this is a possibility as well, if a person is partaking, these alcoholic beverages can also make you sleepy. But as far as the anecdote we were talking about is like, it's the tryptophan plus the sugar, and we get sleepy that way. So just kind of, again, another interesting application. Okay, so now we're at the point where, kind of like with our carbohydrates chapter, right? We said, okay, how do we put these together into larger molecules, right? 2 or 3 or 5 or 10, or a bunch if we're talking about a big polysaccharide. Same thing with amino acids. They can be put into, 2 of them can be put together to form what we call a dipeptide, or a bunch can be put together to form a protein. So that's maybe a good thing to start with. Maybe I'll even put it on this slide. And you'll see different things in different places, but I always like to say, for our class, for us, let's put anywhere from 0 to 50 amino acids, not 0, I guess it would be 2, to 50 amino acids. We're going to call that a peptide. So just think of a peptide as a small protein. Okay, and if we're talking about greater than 50 amino acids put together, right? Not just by themselves, put together, we're going to call that a protein. So peptides and proteins. Okay, so you see, the two amino acids, alanine and serine here, are put together into what we call I guess this would be called a This would be called a dipeptide, right? 3 of them: tripeptide, tetrapeptide, pentapeptide. We don't use that terminology a lot, but I suppose it'd be good to know. So 2 of them, in this case, a dipeptide. What I want to do, though, is oh, what happened there? Oh no, I didn't see that was all. Shoot. Okay. wonder if I can. No, I think I'm just gonna have to rewrite it. Sorry, I was gonna try to grab it, but when I do that, it saves it. So I was gonna grab it with a. So let's just write what we. Okay, so let's put 2 to 50 amino acids. Greater than 50. Protein. Okay, now I want to do this together. In fact, I should have thought I had a. Okay, let me go grab a blank page here. I want to do kind of what's done on this slide, but in a little different way and highlight a few important things. So let me grab another blank page. Let's do it right here. Okay. And I want to say, okay, let's treat. It doesn't really matter. Let's make them generic for now. We'll get more specific here as we go. So let's just say we have. Two generic amino acids. And let's call them R1 and R2 just to make sure they're different, just so we know that they're different. And I'll leave it at that. I'm gonna make the other one expanded, and you're gonna see why we do this. R2, we've done this kind of thing a lot of times before. I'm just trying to show you. Remember, a lot of times we'll pull a water molecule out and we'll say, okay, the result is then those two things coming together. We're going to do that exact same thing here. We're going to take then OH and an H, pull them out, then that new carbonyl to nitrogen bond is formed, and I bet you'll be able to tell me what that functional group is. So what we end up with here is now remember, amino acid 1, this is amino acid number 1, and amino acid number 2. They are coming together to form our now generic dipeptide. Now, a couple things here that are really important to take note of. You kind of got to flip that one upside down to make this look good. Make this look reasonable. Okay, so I bet you'll remember that functional group right there is called an amide functional group. But in this case, so it's an amide functional group or an amide bond, but it's in this case that carbon-nitrogen bond is also called a peptide bond. Okay. Okay. So it's also called a peptide bond, and we'll refer to it as that too. I think I had, yeah. Okay. So there it is right there. It was on that last slide. And those are the bonds that hold proteins together or peptides together. So over and over and over, you have these peptide bonds. Now, one of the really interesting things about those bonds is they're very strong and they're very robust. And there's a reason for that. In fact, it's kind of implied here. Peptide bonds are planar. Now, I want us to think about that together, and we can even like draw this out. Let's redraw this, and let's circle all of the atoms that are actually in the same plane. So I'm going to redraw this generic dipeptide. Yes, R2 here, right? R1 and R2. Okay. And I'm going to circle, I'll use a different color here. Let's use orange here. If you think about this, right? Okay, now, first of all, but before we do this, let me go back to this for a second. This nitrogen has a lone pair of electrons on it. So we're gonna go back to Chem 3A for a second and talk about something called resonance, right? And the idea being with this pair of electrons can actually be pushed over here, and this pair of electrons, remember there's two lone pairs on oxygen here, this pair of electrons is going to be pushed up here. So remember what resonance is. It's all of the atoms are in the exact same place, but they're held together in a different way by all the same electrons, but just in a slightly different way. And so what we end up with here is a situation where we can redraw this. There ends up being a negative charge on oxygen there, and then a positive charge on nitrogen, because it's now attached, as we know, to 4 different things. So of course it's positive now. So there would be a positive charge there. And now remember a few of the important sort of nuances about resonance structures. Probably the most important thing is the molecule on the left, the dipeptide on the left, isn't the exact picture of this molecule, nor is the one on the right. Remember, however many resonance structures you can draw, the real molecule is a weighted average of those. It lies somewhere in between, which is kind of a mysterious thing, but it's all part of, you know, the theory that's the theories, I should say, that we have about how molecules are put together. But one of the things I want to point out, right, is here. Let me go back to this laser pointer. Here you see a double bond, right? Now here there's a single bond and a double bond here, but there's some what we call double bond character associated with this carbon-nitrogen bond because one of its resonance structures has a double bond. Now, it turns out some resonance structures are better than others, and that's why I said a weighted average. The real picture of the molecules, it tends to mimic the better, or I should say be more like the better resonance structures. And we won't go into, that's not something we talk about in 3A or in 3B really, okay, what makes a resonance structure better or worse. But let's just, for our purposes, say that the molecule is somewhere in between here, and it has that amide or peptide bond has some double bond character. Now, here's where we go back to planarity. Okay, so if we have, right, and this is like an aside here. This is like an aside. If we have just a regular old alkene, right, that we're used to seeing, we definitely know there's a double bond right there, right? But if you think about it, right, this is trigonal planar, right? This carbon is trigonal planar, and so is this one. And so really all 6 of those atoms lie in the same plane. This is a planar molecule. So in the same way, if you think about the planarity of an amide, it's important. It's important for a couple of reasons. Number one, it constrains the geometry of the amide functional group, and that's something that later becomes really important when we talk about bigger collections of amino acids, big proteins, and how they're folded together. I wanted to go back and get this orange one, and I wanted to circle the 6 atoms that I guess lie in the same plane. No, you know what? There would be one more. Let's think about this. Yeah, all 6 of these atoms lie in the same plane. Because of the planarity of that double bond. Now, the other thing, so let's say this. Let's say resonance in peptide bonds makes them planar. So A, planar. So peptide bonds have that planarity associated with them, and B makes them stronger. Okay. Again, we won't go into all of the details there, but peptide bonds are, and it wouldn't. Well, we'll just leave it at that. Peptide bonds are very strong, which is again one of the reasons that proteins themselves are very strong, just because they have these repeating peptide bonds. So yeah, so those are good things for us to know as well. Okay. Okay. I always think about Kevlar. Kevlar is a polyamide, and I always think, gosh, Kevlar is one of these materials that we have that we use it on like motorcycle gear, right? We use it in, and I think we talked about Kevlar in our amides chapter, and I think I may have even mentioned, hey, later on when we talk about proteins, what we'll see is that proteins have these amides that are represented over and over and over, right? We have a lot of these really strong bonds, which again translates at the macro level to a stronger molecule. Okay. Now, a couple of things about how. So one of the undoubtedly one of the biggest discoveries in the last 50, 60 years, and it's continued to be a realm where there's been a lot of improvements and things like that, is the synthesis of peptides and proteins. And there's this guy, so solid phase synthesis was pioneered by this guy named, last name Merrifield, and you see part of that original sort of seminal paper here. He synthesized a tripeptide and published it. And again, this was just really profound. I mean, even if you think about some of those peptide hormones that we talked about in that negative feedback pathway, this technology made making those and then using them in studies really a reality. And it turns out, and we won't go into all the details here. In fact, there's so much. Again, this is one of those things where I'd say if you really had a hunger to learn more about peptide synthesis, and you may or may not, we go into gruesome detail, towards the end of our year-long OChem course. But just some of the fundamentals here. Whoops, I thought I had the pointer. And I will say that the way it happens in vitro, meaning in glass or in the lab, is slightly different significantly different, I should say, than the way it happens in a living organism. But basically you have this resin that has, and each of these little beads has tons of different places where an amino acid chain or a peptide or a protein can start. And you have this little molecule we call a linker molecule, and to that linker, which is attached to the resin, attaches the carboxyl group of the amino acid. So there's the amino acid, that's meant to be the R group, and then there's what we call a protecting group, so that the nitrogen doesn't react before we want it to. Well, you remove the protecting group, and then you add it to another amino acid, which happens to be protected also, and needs to be unprotected, and so you add them together, and then this whole process goes on and on and on, and you end up with this synthetic peptide. Now, I guess the thing that there's a lot of detail there, way more even than that's represented in this slide. But I do want you to know this, that peptide synthesis in the lab and I'll just put solid phase. Again, there's even multiple ways of doing it now, but solid phase with a resin, peptide synthesis in the lab occurs from the C carboxyl terminus. That means end terminus, right? To the N amino, right? It's nitrogen amino terminus. So we can at least know that, okay, if nothing else about this, okay. Okay, so this is just, yeah, okay, so this is just a little excerpt. I actually worked for this company a while back, and we made designer peptides and purified them and sent them off to people who were using them in studies and things like that. So, okay, so next thing, peptide synthesis, so in a living organism, in vivo. And here, again, there's so much detail. We'll talk more about this process in our nucleic acids chapter, because it turns out that, right, DNA encodes for RNA, and it's really this RNA or messenger RNA that then is used to make proteins. It's called an mRNA transcript. It's used to make proteins. It encodes for all these different proteins that are linked together by the ribosome, and you kind of see that process here. So again, the details of that, well, we'll leave it at that for now, and we'll come back to some of those details later. But at the very least, we can say, again, peptide synthesis in vivo occurs, I think there's two C's. I think one R. So it's one of those words that you're like, wait, does that look quite right? Occurs the opposite way. So N terminus to C terminus. from the amino end to the carboxyl end. So, and then a new amine is added, amine end first, and so on. Okay. Now, okay, so we're going to now talk again about and we've said this, we looked at a generic dipeptide. So we're going to actually get a little bit more specific now and say, okay, let's say we wanted to actually draw one of these. Now, a couple of the things there, and by the way, C terminus or C terminal end, N terminal end, those are mentioned on this slide as well. We just mentioned them a second ago. But the question is, the thing that we're asked to do here is, can we draw a tripeptide? Namely, one with alanine, serine, and valine linked together in a particular way. Now, particular way here is this, and I really want to, I think I have a, slide here that I'll write this. So let's say this, when writing, I was going to say structures, but I guess structures or shorthand abbreviations for peptides or peptide or protein sequences, right, of amino acids. Okay, we always, or let's say for our purposes, we will always write them, and they mostly are done this way, not always. You'll see them written the opposite way, I should say. But for us, we're going to try to always stick to writing them N terminus to C terminus. Okay, so what we mean by that is, if we're doing that problem we just looked at, the alanine, serine, valine, so for alanine, serine, valine, okay, we're gonna start with the nitrogen of alanine. So we're gonna say this is the N terminus of alanine right here, and this is the C terminus of valine right here. So let's go ahead and write these amino acids, alanine, serine, and valine. Let's write them out, and then we'll kind of put this thing together like we think we should, right? N terminus to C terminus, like we just described. So I guess I can do it. What I expect when you're asked to do this is right away you're going to go to your, one of these sheets we've been looking at of amino acids. I remember what these look like, so I'll go ahead and draw them out myself. And you can, if you want to refer to your sheet, I would encourage you to do that, because just so you can familiarize yourself with those amino acids. Okay, so let's. I was going to draw them out of order just to. I don't know what purpose that would serve. Let me just draw them out as they should. Okay, so. Okay, alanine has a CH3 as its R group. Okay, so there it is. I'll just draw it like that. Serine, if I remember right, looks like this. Yeah, CH2OH, I think. And then valine, again, is this one we've looked at a couple of times already. It's that. I always think valine V for that isopropyl group. So. Now let me do this like we did just a second ago when we drew our generic dipeptide. It is an NH2, but if we draw out the NH2, right, then the nice thing there is that we can kind of excuse me, pull out water molecules and form those bonds like we practice doing so often. So okay, all right, so we've got our, and I'm just going to write them out. Alanine. Now this isn't. Technically, we'd be talking about L-alanine. And by the way, if you go back to the handout that we were just looking at, and I didn't mean for that to be confusing. We know, with certainty that these are three-dimensional molecules, these amino acids, right? And the ones we're talking about are the L versions of those. And so what you'll see is often we'll write the three-dimensional version with dashes and wedges. But what I've done, just to try to simplify this, is I've just written out the two-dimensional versions of these. Just again, we're trying to put these together, so it complicates things a little bit to add stereochemistry right away. So let's not worry about stereochemistry for the time being. We'll just write alanine rather than L-alanine. Same thing here. We'll just write serine. And here we'll write valine. By the way, I didn't leave myself enough room up here, but this could have been written, right? I'll just put ASV or A-S-V. Okay, those are different ways, because remember, this is ALA, A. Those are the abbreviations. I'm making sure. I'm hoping I get these right. I think I'm remembering them right. And this one's VALV. Some of them are weird. They're just not intuitive. These are pretty intuitive. But anyway, so remember, those abbreviations are helpful in that they allow us to write sequences like this, ASV, right? That's a lot easier than writing out alanine serine valine. Okay. So now let's put these together. Let's get our red marker here. And it doesn't matter what hydrogen we choose. Let's just pull a water molecule out. And again, there's a new peptide bond that forms. Same thing here. So if we want to write the whole now tripeptide, excuse me, here's what we end up with. Right? Just everything else goes where it belongs, and we're now forming an amide, right? The peptide bond, as it were, right? There it is right there, peptide bond. Okay, sorry, that carbonyl kind of sucks, but there we go. And notice how when I draw this, the R groups kind of go, they kind of oscillate back and forth between up and down. It's just how the chain is written. So anyway, so there is the next peptide bond or amide, and then the last one, and then here's the next R group, right? Here's our valine R group, and then somehow the scale of this is different than the scale of the amino acids that we drew. But so I want to point out a few things, right? I'll just put peptide bonds, right? Because here is the other one right there. So these are the peptide bonds. And notice how, remember, we set up top there, we want to write these or draw them or represent them with, abbreviations N-terminus. So N-terminus, right? Notice, right, the N-terminus, we're talking about the nitrogen there. And then here is the C-terminus. Whoops, C. Sometimes we call it C-terminal end, right? Same thing, C-terminus. Because this is the C carboxyl group, right? So that's what we're talking about here. Let me just make sure we're in good shape. Yeah, alanine, the methyl group was the R group, serine, the CH2OH, and then valine with our peptide bonds in the middle. All right, looks good. Let's see. Yeah, I guess we have another blank. What's next? Okay, we have another blank page there. Let's go ahead and just because this is good practice, right? You will be, expected to be able to do this kind of thing. Let's do another example. So again, we're just making them up here. Let's do this one. Okay, and I'm just going to give you another tripeptide to work on. Okay, so tyrosine. Let's do methionine and aspartic acid. Let's do those. So if you want, you can pause the video right here. I'm just going to go ahead and do it, and I'll give you the actual. This is so. Let me draw the amino acids over here. So let's see if I can remember these. Tyrosine. I think there's a CH2. Let me draw it a little lower. There's this. I believe it's a CH2 and then a benzene ring. It's like a phenol, right? If we think about it, right? There's an OH there. So those are like a CH2 and then attached to a phenol, and there's the carboxyl end. Okay, so this is tyrosine. And the abbreviations are TYR and Y. So again, it's not TYR and T, right? There's other T amino acids. So, you know, they can't all be T, right, as their one-letter abbreviation. So that's why sometimes you end up with some weird ones. Aspartic acid, one of the other ones that's in this is a weird one too. I think D is the one-letter abbreviation for aspartic acid. So methionine, let me just do aspartic acid next to throw you off, right? To make you put them in the right order. So think about this: CH2, and then there's a carboxyl group. That's weird. Okay, and then another carboxyl group, the regular C-terminal end, and this is aspartic acid. And it's a good reminder, right? Sometimes the R groups are themselves, you know, polar or non-polar, acidic, basic, right? So there's a lot of variety in these amino acids, and that's something we'll talk a lot about towards the end of the chapter. Okay, so ASP I think is the 3-letter abbreviation, and then D, or if I remember right, is the 1-letter abbreviation. Okay, so there's aspartic acid, and then what was the other one we were going to do? Methionine. We're just making this one up for practice. I think there's 2 CH2s and then an S and then like a methyl group as well. So this is actually an interesting, it's a sulfide, not a disulfide, but there's another one of these, I don't know why that looks bad. Another one of these sulfur-containing amino acids. So this would be methionine and MET, and I think it is M as the 1 letter here. Okay, so you've got your amino acids. Now again, if you want to pause the video and practice this on your own, putting it together, I'm just going to go ahead and proceed here. Now remember, as written left to right, we're going to call this N-terminal end, or N-terminus and C-terminus. Now, don't be surprised if you're looking at YouTube videos or whatever, and you end up, you end up seeing something written C-terminus to N-terminus, but we're, again, just for the sake of consistency, so that we always know which end is which, we're going to try to keep it so that whenever we are doing these left to right, we're doing N to C. Okay. All right, so if we put them together, and I guess we could write them like we did before and then circle, and I don't know if that's helpful to you when you're putting these together, but if it is, we'll just continue doing that. So there's our OH. So, and then the next one we said was we're going to do methionine next. So I'll do N and then H and H. All right, CH2, so did I get that right? CH2, CH2S, CH3, yeah. Okay, so there's methionine. And aspartic acid was the other one, right? CH2, carboxyl group, C-terminal end. So then we say, okay, let's circle a water molecule, bring it together right there, circle a water molecule, bring it together right there, and let's draw the whole thing. Okay. Go back to here. And then again, we're going to kind of point the next R group, CH2, CH2, S, CH3, down, right, just because of the way that these chains are written. Remember, we're constrained just kind of by the way the chain goes back and forth. So where am I at? I'm at here's another R group, right? CH2, OH. Okay, so there we are. It looks kind of wonky, this part. Let me see if I can draw that a little bit better. Not really. Okay, there it is. So, so again, N-terminus, C-terminus. We've got 2 peptide bonds. You could identify those as well. There they are. C-terminal end, right? So N-terminus, that's right, and C-terminus. Okay, and then again, just a reminder, this can be written TYR-MET-ASP, or we could write it Y, right? Tyrosine, remember, has a one-letter abbreviation, Y dash, I guess it would be M then, right? Dash D, or sometimes we don't do the dashes, or YMD. Sorry, looks like a YMD. Okay, so these are just ways of writing the sequence, what we call primary protein structure, which we'll get to here in a little bit. Now, so oh no, I didn't mean to erase that. Just draw it in red again. Okay, so what's next? What do we got? Okay, so okay, so it looks like we're about to get to levels of protein structure, and I think that's a good stopping point. Not here, but there. So let's go through the stuff on this slide, and then we'll I think that'll be a nice natural break for the part one of this lecture. So, but first of all, okay. So we referred to this earlier, this idea that an isoelectric point certainly applies to individual amino acids, right? We looked at that table. All of those amino acids have different PIs, right? Remember the pH at which whatever it is we're talking about exists predominantly in its zwitterionic form. Peptides and proteins are no different. So if we're thinking about we'll answer that question here in just a little bit, but if we're thinking about a globular protein, just because it's kind of easier just to draw a big blob, honestly. So let me just draw one, and again, what we're meant to represent here. Okay, let me just get this out here. We'll just start here, and we'll just kind of like make a globule. And what I'm trying to show here is and I wish I could reproduce that like 3 times. In fact, I think I can. So, okay, I'm sort of cheating here. I was going to draw it 3 times, but it wouldn't look the same 3 times. Oh, that's kind of cool. Okay, so let's do it one more time. And you'll see what I'm getting at here, why I'm doing it 3 times. But nice. Okay, so let's go back here. Okay, so the reason I did that was I wanted to show how, again, kind of like we did with the amino acid, at really low pH or at a pH below the PI, the protein exists in this particular form, and if it's above the PI, it exists in this particular form, and if we're at the PI, then it exists in this particular form. And so let me set that up. Go back to here. Actually, maybe we can just keep I need to write a few other things. So let's say globular, we'll just call this globular protein Z, okay? I don't know why. Okay, same thing here. Well, it's the same thing. And we'll just say at a pH below the PI of protein Z. Now, remember, at a low pH, right, a relatively acidic environment, we're talking about a lot of H+. Now, one of the things I want to make sure we're clear about, I'm realizing maybe I didn't make this clear when I described drawing this big, what looks like a ball of yarn, really, right? This is the polypeptide chain, so there's a bunch of amino acids all linked together in a chain, and this thing is all folded up. And again, the folding part, that comes in lecture 2. We'll talk about why that happens. But suffice it to say here, this is a big polypeptide chain, and it's all folded up. Okay, now, if you're thinking about the end, I'm going to try to highlight the N-terminal end. So N, there's our N, and here's our C-terminal end. But let's think about I guess the the form that the amine would be in here, right? So the idea is the amine, right, good base would be, notice, in its protonated form, right? Low pH, lots of H plus. Again, we talked about this earlier. Now, the carboxyl group also would be in its protonated form, right? If it were in a basic environment, yeah, definitely deprotonated. In fact, both of them would be. But remember, we're at a relatively low pH here. So we'll talk about now globular protein. Well, let's jump over here. Well, I think you can probably see where we're going with this. Now, okay, I'm kind of jumping all over the place here. Let me do this just so that we can see these. I'm going to put the positive charges. We'll just do them like. Let's do them blue. Okay, so I'm going to put blue there. Now, we've seen this. In fact, we just saw an amino acid, aspartic acid, right, that had an actual acidic side chain, and there are many of them. If you kind of pour through those amino acids, there are many of them that also have basic side chains. And so I'm just going to draw some positive charges and some maybe I can do negative, a different color. Okay, so some positive charges and some negative charges. But think about this for a second, right? The amines in this, now we're talking about the side chains, right? Determining that we already took care of the termini, right, plural, right? The N terminus and the C terminus. But remember, some of those R groups can also be charged. A very important point here. Let me just do one more plus, right? And maybe I'll do one here as well. And it's just, again, I'm just kind of making up where I'm putting these. And I'll just do a negative charge. I'm going to make it so we can see it. Let's do it in orange. Okay, I'll do a couple of negative charges because, again, this is totally pH dependent. But there you go. And notice how there are a lot more positive charges at low pH because, again, those nitrogens would be protonated and you'd end up with formal positive charges. Now, it is true that at whatever pH we're talking about, there might be a couple of negative charges, but there is a net positive charge on the protein, and you could write that, or we can write that underneath here when we're done as well. Okay, now let's jump back to, we'll get to this virionic form here in a second, but let's compare and contrast this with the other two, or with this one, right? So we'll put at pH above pi, significantly above pi of protein Z, right? Remember, the opposite would be true. In this environment, right, there would be not a net of hydrogen, H+, there would be more hydroxide, right? And so it's a basic environment, and the OH minuses steal away H's from both carboxyl groups and ammonium species, right? Like the one we just drew, the nitrogen that was actually protonated. And so you would actually have a net of negative charges here, right? Or the protein would be net negatively charged. So let's write that. In fact, let's write, since we have our black pen here, right? This is not any longer in the NH3 form. It's minus a hydrogen, so it's in its neutral amine form. And the carboxyl group, right, has lost its H, right, in a basic environment at a higher, relatively high pH. Now let's put our positives and negatives here in, and in this case, right, the idea is we can keep a couple of those positives, right, maybe that one and that one, but definitely we're gonna, whoops, definitely we're gonna have a lot more of the negative, so we'll keep the ones we drew here, and we'll just add a whole bunch more, or a number of other ones. Okay. Why is this happening? There we go. And I meant to draw this one just with the, oh shoot, it's saved already, it auto-saved, so I'm just going to draw over it. We'll call that a negative charge as well. Okay, so net negatively charged at relatively high pH. Okay, now, if we're talking about the globular protein Z, we'll just put at pi, okay, right? So the pH at which the zwitterionic form predominates, right? What we end up, remember the things that are true about zwitterions. They have a positive and a negative within the same molecule, and in this case, this is a big molecule. It's not just one amino acid, it's a bunch of them. So the idea here is that we actually have a bunch of positives and negatives, but in the end, we will have the same number of positives as negatives, right? Because remember, the net charge, the net charge on a zwitterion is, well, they're neutral, right? So same number of positives as negatives. And I wanted to make those blue, right? The positives. So I'm going to put the amine in its ammonium form, so its positive form, and then the carboxyl group in its, again, I'm going to put that in orange, in its negative form, and we'll have the same number of positives as negatives. So just to make sure we do that, so 1, 2, 3, maybe one more here, and then we'll do the same number of negatives with negative there, and then we'll do a negative there. How many did we do? Negative there, negative there, negative here. Okay, so we can write a few more notes. So here, right, we have a net charge, obviously positive, right? Net charge negative, and here net charge Na, right? It's neutral. So one of the things, and back to this for a second, just to set this up, and then we can kind of make some conclusions that are on that slide, is that proteins and enzymes in particular have just like all these other small molecules we've been looking at, have, you know, relative solubilities and I guess melting points and boiling points. We're going to focus on solubility here because sometimes, and we'll get to this here in a second, but sometimes it's helpful to bring up one of these molecules out of solution because we want to study it in its solid form. Okay, but if you think back, right, to where we were just at, and here's the question: why is the protein least soluble at its isoelectric point? I guess we hadn't even said that yet, but it's true. So at its isoelectric point, the protein or peptide that we're talking about is least soluble. Okay, why is that true? The fact that is true, we're saying that now, but why is it true? Now let's think about this for a second. Back to Chem 3A and things we know about ionic compounds and solubility, right? Ionic compounds, not every single one of them, but lots of them are relatively water soluble, and it's really because of those interactions between charges and water. We call those ion dipole interactions. They're really polar intermolecular interactions, that things that are charged are often surrounded by water molecules and brought into solution, so they're very soluble for that reason. So what we're saying here is for that reason, right, this version of protein, in fact, we can say that this version, right, i.e. the zwitterionic form, of protein Z is least, right, charged overall. Now it still has charges, but they cancel out, and also least soluble. Now remember, increased charge often corresponds to increased solubility, not always, but for ionic compounds. Okay, so that's really the reason here. Another thing is, so there's two issues. So I'll just put, if we're thinking about solubility, this is one of those, and this isn't laid out very well, but I'll just put this as well. Also also the I'll just put the zwitterionic form of proteins, and this is all kind of linked together conceptually, but proteins tend to aggregate more if they're in their zwitterionic form this further decreases solubility. So really two issues here. They're actually, again, theoretically, the theory surrounding this is it really permeates both. It's part of the reason. Well, they're both linked to the same theory is what I'm getting at here. So, but both of those things I want you to know, right? So overall less charged equals lower relative solubility, and the aggregation makes these aggregates are really big, and when molecules get really big, especially if they have a lot of nonpolar things, they're also less soluble for that reason. So, okay, now why do we care? And that was the other thing that was part of this question that we were asking ourselves. And it's really just, again, another one of these kind of interesting applications. I'll bet some of you maybe have heard of Staphylococcus, or it's a group of bacteria, and one of them in particular, or some of them, are really resistant to antibiotics. So you may have heard of MRSA, methicillin-resistant Staph aureus, and it's one of these scourges in, well, lots of places, but one of those places is in hospitals, because it's hard to kill it. It wants to persist, and it's really troublesome if you get infected. If it's part of an infection, and so I guess what I'm getting at is if we can study proteins. In fact, you see one here on the slide. This is a protein that MRSA possesses, and it's thought to be one of the things that makes it able to kind of persist and be virulent and do the bad stuff that it does. And so you could imagine, okay, if we could take some MRSA, and this there's a lot that goes into this, but break it open, harvest some of its proteins, and isolate this one in particular. Again, a lot of work goes into that, but these things can be done. Then if we could take that molecule and bring it out, if we had enough of it, we could bring it out of solution, because it would exist in solution if we were to isolate it, bring it out of solution, make it into a solid, cause it to really crystallize out of solution. And think about crystallization or precipitation. And again, this is all altering solubility and maybe even the pH of the environment that the protein is in to make it come out of solution. You see this picture on the right here. It's of a what we call a crystallography instrument, or an X-ray diffractometer. And what happens is, and I think we've discussed this before, like it seems like when we discussed benzene we talked about this, but if you have a really small amount of really pure solid, crystalline solid, you could mount it on that probe. In fact, let me get the laser pointer. There's a little probe here, and there's a lot of things going on. This is like a camera, an X-ray detector, and there's cryogen gases being shot on the mounted crystal just to cool it down, to make sure there's less molecular vibration, and then you shoot X-rays at it, and you can actually calculate. There's a lot of complex calculations associated with all of this, but you can backwards use the data to determine what atoms are there, what amino acids make up this protein, and then and you can actually use these models to develop drugs. Pharmaceutical companies do this all the time, and there have been people who have made a whole career out of crystallizing a particular important protein. And so again, those things are often related to solubility and pH and things that we've just discussed. So really interesting stuff, really cool stuff. Okay, so let's see here. All right, so let's call it good for part 1 of our chapter 14 discussion.